Yeah, in general, Pseudomonas, they don't, except for Pseudomonas aeruginosa, which can cause some lung issues, most of them don't have true like pathogenicity. Most of them don't have like an inherent ability to cause disease in people, right? Unless you just eat too much of them and that upsets your body. We really think of those Pseudomonas more as spoilage organisms. So also bad, but something is going to spoil meat at some point, no matter what. So it's not like they're some mortal enemy we have to fight. We just want to deal with them to extend our shelf life. And so.
Welcome to MeatsPad. It's a platform dedicated to share breakthrough knowledge that is accessible to the global meat industry. On each episode, you'll have the opportunity to listen and learn from meat specialists and professionals about the whole meat industry. This podcast is brought to you by Ultrasource, equipment and supplies for the meat and food industry.
Hello, meat folks. Welcome back to the MeatsPad podcast. It's your happy hunger host, Phil Bass. Guys, we have a unique situation today. Never have I had two generations on the MeatsPad podcast, but today is we are crossing that one off the list. A lot of listeners, if you've been listening closely, will know that for my PhD, my advisor and mentor was Dr. Keith Belk at Colorado State University. Well, we have another Dr. Belk on the line today, Dr. Aeriel Belk. And she is at Auburn University, and yes, a close relationship to Dr. Keith Belk. We'll get into that later on, but Dr. Aeriel, thanks for joining us today.
Oh, thanks for having me. It's always fun to be a record breaker. So
yeah, how about that? So yes, definitely we'll kind of dig a little bit into how you came about to becoming Dr. Aeriel Belk. But the reason I wanted to have you on is actually you've been investigating an area of science that has become it's not a centralized topic of interest to me, but is definitely of interest to me because in the past I've done dry age work and has led me into this world of the microbiome. And so Aeriel, can you maybe let's lay the groundwork for the listeners and describe what is a microbiome?
Oh, well, that is an excellent question. I think different people will tell you different things, but from my perspective, a microbiome is all of the microbes in a community in an environment. So in meat science, a lot of times we're looking for specific organisms, right? We're counting the number of E. coli in a sample. But with the microbiome, we're looking at everything that's present, not just our one target, and we're looking at it by using the DNA. So usually some sort of DNA sequencing mechanism to figure out what organisms are there, which is helpful because it lets us capture the organisms that are non-culturable or not yet culturable, as some people will say.
Right. So and that's what really has drawn me into this whole discussion, this whole topic is that as you mentioned, it's a community, right? And so for the longest time, we have intensively focused on specific microorganisms, but what I have experienced from the dry aging and quality side of meat science is that some of those bugs they like what they like, and they like the other organisms that they like, and will create kind of their own community. I mean, think of it as just a small town, and maybe you have the Belks, you have the Basses, you have the Earps, and you have the Hatfields and the McCoys and the different folks that make up that community, and some get along with others different. Tell me a little bit about how this is starting to be further investigated, say, in the meat sciences, and maybe tell us a little bit more about those microbiome communities.
Sure. Well, there are a lot of microbiome communities that are relevant to animal science, everything from those associated with the live animal to those associated with people who are eating the meat, and then kind of everything that happens in between those two parts, right? So I think the microbiome research really entered animal science as a broader field through like rumen microbiomes, right? Because that lets us really understand a lot more about how the digestive process works. And they're pretty easy to study because we can get the cannulated cattle and pull out a sample. And then those are also connected to meat safety, because the more you disrupt the native microbiome of a animal, the more likely you are to have a different microbiome or pathogen load in the meat, right? So that's where it starts. But we're looking more into the actual microbiome of food products themselves now. And a lot of people are investigating, like you've mentioned, the microbiomes associated with dry and wet aging.
Hmm.
Different microbiomes relate to that. And also in the processed meats area, the microbiome of like fermented meats, right?
Hmm. Sure.
People know that there's a microbiome in there. You may have a historic one, you may have one that you're introducing yourself. But that community of microorganisms in there and how they interact is really important to how that product is going to taste.
Right. Yeah.
From my side, trying to connect it to food safety, I'm looking at the microbes involved in meat spoilage, which is like the other side of the coin of aging, right? But how
Right.
microbial communities change over time, how competition of microbes within that community may slow or speed spoilage, and then how those may interfere with pathogens that are present during that spoilage period.
Right.
And then I'm also really interested in the microbiome associated with the built environment, the human-made structures in which humans live and work and otherwise occupy. These also have distinct microbiomes, and that includes our meat processing facilities. So how does the microbiome present in a meat processing facility influence the survival and persistence of pathogens? A lot of P's to say on a microphone, so sorry about that.
Well,
That then can contaminate food.
right. Well, let's expand upon that. What are some of the findings that you're discovering? And I'm thinking a little bit about so for those listening, especially AMSA, American Meat Science Association members, you can go and listen to one of Dr. Belk's discussions on the latest reciprocal meat conference proceedings. But it was really fascinating to see just how unique some of these environments are to unique structures, unique premises and things like that. So let's expand a little bit on that and things for our meat processors to think about with regards to, as you said, the built environment.
Yes. So the only project I have completed that's led by me is from my PhD research, where we were tracking how the microbiome of a facility originates and becomes stable over time in a new meat processing facility.
Mhm.
I also did my PhD at Colorado State. I was under Jessica Metcalf there, who does a lot of microbiome research in all sorts of fields. And so we opened the new meat lab at Colorado State, and we took samples of the microbiome every month for 18 months, beginning with like post-construction clean all the way through. And our results got a little wonky because COVID happened there in the middle, and that threw off some of the production systems. But what we could see is it went from basically no microbiome because of all of the intense sanitation to developing kind of distinct microbial communities that really depended on the use and conditions of that space, right? So you might, kind of like you might expect, the animal holding pens and the harvest associated rooms had really similar microbiomes to the animals. And we actually did a little bit of a statistical analysis to show that most of the origin of that microbiome was the animals that came in.
Mhm.
And and it was a really diverse community there because of all of the different incoming sources, right? But then by the time you make it into the fabrication rooms and the coolers and the processing rooms, the microbiome is much less diverse. It was basically just different strains of Pseudomonas at that point,
Mhm.
probably because, we do such a good job removing all of those animal microbes from the meat before it gets into the coolers. Pseudomonas in that type of organism, they're pretty environmental, so they can get brought in and through air, things like that.
Mhm.
Much more difficult to control than some of the animal associated ones. And most importantly, those rooms are kept cold. So you're really selecting for psychotrophs in those, or psychrophiles in those spaces.
Mhm.
And what I think is important about that is we're really learning how the way you design your facility in terms of physical space and temperature and everything else, where people are allowed to go, whether you have to change clothes, the PPE you wear,
Mhm.
really impacts what's going to be residing in your facility. And what resides in your facility can then have major impacts on ultimately what your food looks like.
Right.
We were collecting just from drains, which don't necessarily come in contact with foods, but are pretty good representatives of what's in the facility, since everything ends up going down in the drains. That's also, where you see the biofilms start to form that may help those organisms persist. Yeah.
So you mentioned a few microorganisms, and I love to kind of call a few of these out, and especially you talked about Pseudomonas. For those who are not in the scientific community and less familiar with it, tell us a little bit about Pseudomonas. Is it a good guy? Is it a bad guy? Is it just a guy we got to deal with?
You know, there's a certain philosophy that says all microbes are just guys we have to deal with and they can be good and be bad, right? There's this whole philosophy of even what is a pathogen, and it has
Right.
to do with, well, how susceptible is the host at that point, right? But
Right.
yeah, in general, Pseudomonas, they don't, except for Pseudomonas aeruginosa, which can cause some lung issues, most of them don't have true like pathogenicity. Most of them don't have like an inherent ability to cause disease in people,
Mhm.
right? Unless you just eat too much of them and that upsets your body.
Right.
We really think of those Pseudomonas more as spoilage organisms. So also bad, but something is going to spoil meat at some point, no matter what. So it's not
Right.
like they're some mortal enemy we have to fight. We just want
Right.
to deal with them to extend our shelf life and so on. But yeah, so Pseudomonas are organisms, they need air to live and they need cold temperatures to live. And then also, lots of moisture, lots of nutrients. And so that means meat in a refrigerator is a really fantastic environment for them to grow, right? And some Pseudomonas specifically are especially good at growing on meat because they have the ability to kind of break open some of those proteins, break them down further to use them when other bacteria don't. So they're able to kind of compete and live in that environment, but also kind of feed other bacteria that are there as well, which is again why the community is important instead of just saying how many Pseudomonas there are.
Right. Yeah,
That,
yeah. So seeing how they interact, but yeah, I mean, I'm glad that we talked a little bit about the pseudomonads. They're back to some of my dry aging work. They're very prevalent because again, we're just exposing meat to a refrigerated environment. I mean, yeah, they're everywhere. They're everywhere, but doesn't necessarily mean it's a bad thing. So wanted to make sure that folks out there are familiar with that anyway.
that is such a good call because I do feel, I mean, you need to be careful doing this type of work, right? That you
Mhm.
want going in and you're going to ruin somebody's day by telling them they have pathogens at their facility. So we do have to be a little careful.
Sure.
But I should also say, like Pseudomonas, they're the main one we look at as a spoilage organism in like aerobically stored
Mhm.
food that has the availability of oxygen. When you switch to like a vacuum package situation, you get a lot more lactic acid bacteria as your main drivers. And those can also be found sometimes in facility microbiomes. It's just a little harder because they require no oxygen. And usually there's a lot of oxygen available in like a room because
Mhm.
we need it too. So.
Right, right. So you mentioned that pseudomonads are one that will likely just travel in through the air. What are some of the other microorganisms that you found, and how are they getting into some of these environments? And is that something that we really need to intensely focus on, or again just manage?
Since 1883, Ultrasource has been a trusted supplier to the food industry. Ultrasource provides superior kill floor, processing, packaging, and labeling equipment and operational supplies.
Well, that is a fantastic question, and it's still kind of a ongoing question. I haven't tracked it all down yet. When we were conducting our study, we looked at some of the different potential sources of microbes into the facility, and you could explain a lot of them coming in from animals, from people, from like out soil that's outside that may get kind of trapped in and carried even when you change boots.
Mhm.
Um, but a lot of it we couldn't quite explain yet. So I'm trying in kind of some studies I'm starting now to look at more air sources that are coming in because that's my best guess for how they're being transported. Uh,
Mhm.
but at a certain point, it's the idea is they just get colonized once and they find a way to kind of persist in the environment somehow. And so even if you are very careful, if you've let them in, you got to keep watch, or you still have to watch for it. So I think in terms of concern, facilities usually do a pretty good job trying to prevent organisms from being brought in, right?
Mhm.
Have your PPE, you wash your hands. I think the bigger concern really should be on kind of sanitizing, cleaning, and especially removing biofilms,
Mhm.
which is a large way that organisms can stay in those microbial communities,
Mhm.
even if you sanitize every day. So I think that's the side to fight more than necessarily what's coming in right now.
Can you expand just a little bit more on the biofilms, especially for those maybe who are, you do a good hot water rinse and hot water that should handle everything, right? But the reality is that some of these guys are able to these little microbes are able to hold hands and stick around for a little bit longer, right? And
Well, I was
philosophically speaking, they don't have hands, but
making little like igloos or something that they
yeah.
like up in, right?
Yeah.
Some microbes, many microbes in food systems are able to make it's basically like a combination of sugar and protein that turns into like a glue, right? So they can make themselves happy little houses that they can stick on to, the drains, the walls, the conveyor belts, whatever else you have in your facility. Even on microscopic levels, even though it seems like this sort of structure you should be able to see, you can't always. And they can then persist inside of this. And what's more troubling from my perspective is they can then recruit or allow other organisms that are different species to come into it. So you have this within your built environment microbiome, you have your biofilm microbiome, right? All of these organisms kind of growing together. So this allows organisms that aren't as good at making biofilms like Listeria to survive inside of them. Yeah, the best way to think about biofilms is like, that's what plaque is on your teeth. It's a biofilm, right? So when your dentist has to scrape it off or your dental hygienist scrapes it off, that's removing biofilms. And we need to do that same thing in our facilities. And people are getting, from what I understand, the people I've talked to, what I understand, better at approaching that. There's even some kind of chemicals that are supposed to be pretty helpful to stripping biofilms chemically. Generally, scrubbing does a pretty good job. We just have to remember to do it.
Yeah, don't forget to that old-fashioned elbow grease goes an awful long way when it comes to cleanliness and sanitation in our plants. Just because you can't see it doesn't mean it's not there. And so keep an eye out. And I think that's really important that you mentioned that, we may have just a mundane group of bacteria that have established a biofilm, but like you mentioned, they can recruit, they can bring in others that maybe they'll get along with. And now you have potentially dangerous ones that are intermingling.
And that's the research I'm working on now. I got some USDA money to look at this. So what we're doing now is growing biofilms intentionally in small meat processing facilities on little like stainless steel coupons. Looks like a slide, except made of steel. And so we're using that to grow microbiomes to see what they would actually look like in the facility, pulling them out, taking them into the lab, and then in the lab, exposing them to Listeria monocytogenes. With the idea is we can see first a diversity of microbial biofilms. How different are they in different facilities? Or is there kind of one meat processing microbiome that forms? We don't know that yet, so we're working on that. And then if there are different communities, is one more or less likely to let Listeria join the biofilm? And so with that, I'm hoping we can figure out a biofilm microbiome. So that is like an early warning sign for Listeria. I could go into your plant, sequence your microbiome, tell you what risk you are for developing a Listeria problem. And then the flip side is, are there some organisms we can maybe seed into processing facilities that can help prevent Listeria growth? Is there something that will generally outcompete Listeria in those biofilms? Some work done by colleagues of mine, so like Jasna Kovac at Penn State University found some organisms like certain Flavobacteriaceae that will, in like artificial biofilms, decrease Listeria's ability to persist in there. So I'm hoping we can find something like that that's already kind of naturally occurring, or naturally, if you want to, artificially occurring in facilities.
That is so cool. That, so often, especially in the meat safety world, we're trying to eliminate. But if we completely eliminate, and this is what I talk about in my class, you can correct me if I'm wrong, but this is what I talk about in class, is that if we make it so that a pathogen could just go there and they have no other competition, there's a good chance they're going to set up shop.
Exactly, yes.
Right.
I think we have historically ignored, well, maybe not ignored, but underutilized the fact that microbes are always in warfare with each other. Your Hatfield his metaphor there was really apt, right? In general, the microbes, they want to survive and they're out competing everybody else in that community via different methods. And we can take advantage of that. I mean, and we have, this is why, you know, like say people tell you to eat yogurt after you had antibiotics, right? To try to reseed that microbiome. This is why if you take a lot of antibiotics, you can get suddenly something like Clostridium difficile or C. diff, you may have heard, or that way, like might attack you and make you sick when it never did before, because now it can grow unchecked. And then you need a fecal transplant to fix you, right? And it's the same thing in environments.
Right. Yeah, using these native bacteria to our advantage, and I like how you mentioned yogurt. You know, it's extremely popular now to have live culture yogurts in the grocery stores. You know, yeah, it's totally beneficial.
And whether you can actually, I mean, breaking into the human health side of it is a whole long thing, but I just want to. Probiotics are a good thing, especially if you've done something that might have depleted your gut microbiome, but they are not like a fix it for everything as they're sometimes marketed as. And I do just, you know, if I'm being recorded, I want to make sure I don't sound like I'm saying it'll fix everything problem you've ever had. But there's definitely value to it, and that's definitely something that is useful to incorporate into our foods.
Absolutely. Yeah. So yeah. Back to the point of microbes. Are they good? Are they bad? Are they just a guy we're going to deal with, work along with? And there's so many beneficial ones. There's even more that, you know, they're just there and they're just hanging out. And I love that your research right now, and I'm excited to hear how your current research turns out. But I love how you're looking at the bigger picture, and it's not just the one guy and the one bad one, or we're looking at the greater community. Are there ones that they're going to interact with better with? Are there ones that they don't get along with, but could be beneficial to us? I think that's a great direction from a food safety standpoint.
Well, thank you. It's really fun. It's cool to dive into this whole environment that no one's really looked at before. It's like discovering a new like forest and seeing all these new animals and seeing who's the predator and who's not, you know?
It really is. So I like to ask folks who join us for the first time anyway on MeatsPad, how'd you get to where you are? You're currently at Auburn University. You come from a very strong meat science tradition. Tell us a little bit about your background.
So as you mentioned, I kind of grew up in the field, the industry. My dad didn't want me to go into meat science. He wanted me to be a doctor. So it wasn't like he was bringing me along to RMCs as a kid, like some people's stories, which is unfortunate now. For me, that would have been a better story, right? But you know, I grew up liking science, liking teaching a lot, liking writing. And as I kind of went through high school, was learning all the different science things, what I really loved was DNA. And so I was like, great, I'm going to go to college. I'm going to be I'm going to study beef genetics. I'm going to look at DNA. And then in college at Colorado State, I started working in the meat microbiology lab. I was on the meat judging team. I did meat science quiz bowl and went to RMC. And I realized, one, I loved the microbes more than I loved PCR. And two, I really liked the meat community, right? And I went to the RMCs and I saw what a community we had in the Reciprocal Meat Science Conference, and saw the community we had and how everybody was like a family and supported each other and knew everybody. And I wanted to be a part of that. So then I went to grad school for meat science, did a master's at Texas A&M, looked at salmonella lymph nodes, really liked the traditional microbiology, but then had the opportunity to go back and do my PhD at Colorado State, and using the microbiome to investigate different questions related to meat and decomposition. And it really became the synthesis of everything I loved, right? Because I was doing bacteria and safety, but I also got to do DNA because remember, we study microbiomes by looking at bacterial DNA. So it was like, I don't know, perfect, meant to be, and loved it. I love how flexible it can be. Once you understand the microbiome, you can work in a lot of different fields, a lot of different research questions. You never get bored. Really enjoyed that. When I got after I got my PhD, I did a brief 1-year postdoc at the University of Maryland, and then this job at Auburn opened up, and just to come work in meat science, be on faculty here. And so I applied for it thinking I only have 1 year postdoc experience. I don't know if I'm a little scared to move to the South, but it'll be good interview experience if nothing else. And then the second I got down here for my interview, I was like, oh no, I love it here. I really want this job now. And it turns out they liked me too. So it all worked out. And now I'm here at Auburn, learned to live with the humidity and how to tell which version of Bless Your Heart is a nice one. And pretty much sticking around. So
Yeah.
building a lab, helping rebuild our meat judging team, enjoying working with different faculty here that I can collaborate with. And it's really feels like everything kind of just came together, you know?
Yeah.
So
Well, that is so cool. And I love the story, where you didn't have the intention of going into meat science, but, it found you. And we're grateful to have you as part of the American Meat Science Association family. I'm grateful to have you on the podcast. Let's keep talking as your research progresses. I'm really excited to see where this takes us.
thank you so much. It was a delight to be here.
Oh yeah, yeah. Well, thanks a lot, and everyone, make sure you tune in next time. Take care.